So now I'm going to present the major application of a software for ultimate understanding, explanation, and presentation of all kinds of multiomics data. Here I have used my own software to present the deep analysis of any kinds of multiomics data, and it almost covers the entire application of computational systems biology and bioinformatics. And this paper, this software was published in 2022, and it has been included here as a screenshot for the paper if someone wants to visit the paper and read it. So here I have just included gene expression or mutational things, but you can include all kinds of multiomics data, whether it is mutational profiling, epigenetics kind of things, or also proteomics data, ataxic data, GECIP data, single cell sequencing data, any kinds of data. And the software could also be implemented for selected genes or proteins. If you have the genes or proteins list, you can execute this software for in-depth analysis. This software replaces the this is the complete replacement for Ingenuity software, which is highly expensive and highly commercial software. So let's start. The workflow for this software is, if you have raw data for any kinds of data, gene expression, mutation, proteomics, or any kinds of data, then go for the prediction or preparation of the important genes or proteins. For example, here it is differentially expressed genes or DEX, and then predict the pathways. That is called enriched pathways. And after that, there is second step. So basically, all these things are first step. Now, in the second step, you need to prepare the network of genes and proteins, genes or proteins, and go for calculation of many other parameters, such as genes shared between the pathways, genes present in the pathways, number of links for each gene, total number of links present there. And this you can predict by taking the help of any kind of network databases. And in the final step, you can go for the calculation of interactions between signaling pathways. So basically, one signaling pathway is interacting with another, or another with another one. So hundreds of signaling pathway could interact here. This is called pathway crosstalk. So this is summarized workflow. Now, if you go for more summary, you can see this workflow. Like, let's say, down-regulated genes, up-regulated genes, or differentially expressed genes, go for enriched pathways, calculate the crosstalk degree, then predict the classes, for example, pathway-pathway interaction, and the corresponding genes. If you are wondering if it is something completely different, then I'm presenting some relevant workflow for the previous existing softwares. And these are the generalized workflow for most of these softwares. So next is, here it is one of the most important database after the patient samples or data, like genomic data or proteomics data and network databases. This is third one, which is most important thing, that is called pathway database. And here we have included cake pathways. Cake pathways have 7 categories of pathways. First category is basically, let's say, cellular processes. Second category is genetic information processing. Third one is metabolism. Fourth one is environmental information processing. Fifth one is organismal systems. Sixth is human diseases. Seventh is drug development. So the drug development pathways are not any dedicated pathway. It is just the modification of the pathway categories belonging to metabolism or these 5 pathways. Similarly, human diseases are in the same way. For example, prostate cancer pathway, human cancer pathway, or many more things. So originally, these are the 5 categories of pathways, which are mainly the real signaling pathways categories. And then each pathway categories contains a large number of pathways. So you can see I have utilized these 5 categories only for the software which I have developed. And these categories are now here, which I have utilized for the software. So now, cellular processes, environmental information processing, genetic information processing, metabolism, and organism systems. This is process to process or pathway to pathway crosstalk or interactions. Now, this is one of the sample for the crosstalk outcome for prostate cancer when we are utilizing these 5 categories of pathways only. And this is called CTA score, that means crosstalk score 0.1, CTA score 0.2, CTA score 0.3, CTA scores higher than 0.5, CTA score higher than 0.7. So if you will try to understand, these numbers are representing the pathways names, which you will be seeing in the next figures. But this is just each circle is basically, or node is representing the pathway. And thicker the connectivity between these nodes or pathways, that means stronger the interaction. Thinner means weaker crosstalk. So this is what we are highlighting. Now, if you want to see how it looks like overall results from gene expression analysis processing to pathway analysis,